In this lecture, we're going to explore the concept of how signals get across the membrane, and we'll draw many common themes that we've seen so far, including binding interactions and conformational change. We'll also look at what happens when there's a problem in the pathway, so things like cancer are extremely susceptible to any dysregulation of signaling. Our cells are held in a delicate balance between growth and destruction, and if growth isn't regulated, that's when you can get tumors growing. We'll look at the general features of signaling pathways, and you'll see that it really is an amplification process. A single hormone can bind and produce many intracellular secondary messengers that can activate lots of other proteins in the cell. We'll look at the GPCR signaling pathway, which you may have covered in your other courses, and also look at the role of phospholipids in signaling. Some of the membrane protein receptors, and we are focusing on events that are happening at the membrane, they're also enzymes, and we'll see what happens when they get activated. And as I mentioned, we'll also see a few implications of the regulation of these pathways for our overall health. Just a few supplemental readings to help supplement your learning. When it comes to signaling, we're going to focus on the non-steroidal hormones and how they elicit responses through membrane protein receptors. Cells need to be able to communicate in order to coordinate all of the biochemical and physiological processes in the body. And they also need to have the ability to adapt to changes. So things like producing energy or digesting food, the transport of metabolites, or cell growth and division. When a cell is told what to do through some sort of external stimuli, it leads to the activation or inactivation of metabolic pathways or gene expression inside the cell. And this is all going to be mediated by proteins, whether they be enzymes or transcription factors. So what are these signaling molecules? They're typically chemicals, usually released from peripheral or neural cells in response to some stimulus. So say you're out for your weekly grocery shop and you just saw the last bag of flour on the shelf. There's signaling that's going to be occurring at multiple levels, from seeing and recognizing that prized purchase on the shelf, to generating energy and making your legs move so that you can snatch it up before someone else gets it. These signaling molecules can be small ions or sugars, and we can also have hormones that either act across huge distances in the body or more locally on cells around them. Hormones can be steroid derivatives, so derivatives of cholesterol, but we're not really going to focus on these. What we will look at are amino acid derivatives or polypeptide hormones that bind on the outside of a cell. When these molecules bind and initiate a signaling pathway, it's really important that we can turn them off, and this would be known as feedback mechanisms, so that we don't see that uncontrolled signaling leading to disease. Signal transduction refers to transmitting the information, and we do call it a cascade as there is quite a bit of amplification that can occur. This means that as a signaling molecule binds to a receptor, the message is relayed across the membrane, usually through a conformational change in the protein. But the conformational change can also activate a domain that might have some function, like an enzyme. The change in structure on the inside and activation leads to multiple responses on the inside. So the downstream events are things that are happening inside the cell, and these will amplify the signal and lead to things like gene expression or the activation of other enzymes. These signaling pathways are complicated maps, and the stimulus really does need to be specific for a certain receptor. The signaling molecule that binds on the outside would be considered a primary messenger, while any chemical molecules that are released or produced inside the cell would be considered a secondary messenger. Once the cell has responded, it does need to have the ability to turn off this cascade, and this will need to happen at multiple levels, such that the signaling molecule could either unbind, or you could have internalization of that membrane protein receptor, or maybe the inactivation of the enzymes or breakdown of those secondary messengers inside the cell. GPCRs, or G-protein coupled receptors, are a large family of membrane proteins, and they actually make up 35% of all known drug targets. So drugs can either mimic substrate binding if it's an enzyme, or compete for binding to receptors like the GPCRs. These membrane proteins have similar structure to bacterial rhodopsin, which we looked at in the membrane protein structure lecture, in that they consist of 7 transmembrane segments that cross the bilayer. There is a binding site on the extracellular surface of the protein, and binding leads to a conformational change on the inside that releases a G protein. They can bind to many different molecules, and one example that you may have heard about is epinephrine, which you can see on the right and is also an amino acid derivative. Can you figure out which one it is? Isoproterenol is an epinephrine derivative where the addition of two more methyl groups can actually result in a stronger affinity, and this can also activate the same receptor, which is known as the beta-adrenergic receptor. So you can see that they have similar structures, so this phenyl ring with some hydroxyls, so you might have some hydrogen bonding going on there as well as some hydrophobic interactions, and there might be some other hydrogen bonding or ionic interactions with this hydroxyl or amine, and maybe even more van der Waals with those extra methyl groups at the end. The KD values are shown on the right, and these measure the binding affinity, where a lower KD means a stronger interaction. I just want to talk about binding again. I know I've been harping on it quite a bit, but do remember that binding really means forming these non-covalent interactions. And this top view on the right shows 3 of the TMs of the beta-adrenergic receptor and the interactions that occur between the hormone and the amino acid side chains. So we can see the hydrogen bonds forming with serine and asparagine side chains, and there's also a salt bridge that'll form between aspartate and the amine. And you can get ring stacking with a phenylalanine as well as other van der Waals interactions allowing these molecules to bind. The binding affinity can tell you how strongly something interacts, as well as whether or not binding is general or more specific. And these KD values, or dissociation constants, are a function of how much free molecule and how much binding target you have, so the concentration of A and B, divided by the concentration of bound molecule AB. You may also have heard of association constants, or Ka values, but don't get these confused with acid dissociation constants, which we've looked at before. The association constant is just the inverse of the dissociation constant, and we can also use the rate constants for forming the bound complex, or K-on, and the off rate, K-off, to determine these values. Now, I'm just providing this information for your knowledge. Really what is important is that you understand how things bind via non-covalent interactions and how we can characterize binding by measuring these KD values. We've already seen that with transporters, they can be saturable and can only transport as many molecules as there are binding sites. This is referred to as stoichiometry and is obviously dependent on the protein structure, and the same goes for hormone binding. And when it is a non-covalent interaction, keep in mind that this is a dynamic process and that binding is reversible. Molecules don't stay glued together unless there is a covalent bond present. So once the hormone binds, we do need to get the signal across the membrane or activate the G protein that's on the inside. Brian Kabilka was also very much interested in the structure of membrane proteins and probably took 20 to 25 years trying to get snapshots of what these proteins look like. He was studying the beta-2 adrenergic receptor and was trying to see what the protein looks like both in the on and off state so that he can compare what happens when ligand binds. So we've already seen how epinephrine binds on the outside to TM3, 5, and 6, and he did see small changes in TM5 on the extracellular side. So on the right in gray is what the membrane protein looks like without ligand, and then when it comes in and binds, represented in yellow, we see a very subtle change when it's activated, and this is shown in the green ribbon structure. So we've overlaid the two structures so that you can see the before and after. Binding induces this very small movement around the ligand, but this translates to a larger movement in the adjacent TM, TM6, which favors G-alpha activation. It's this 14 angstrom movement at the base of TM6, which would be on the cytosolic face of the membrane, that can transmit the signal across. In 2012, Brian Kabilka and Robert Lickowitz were awarded the Nobel Prize, not just for this structure, but for understanding how hormones bind and elicit these types of responses. Now, we've talked about in general how you can get these types of structures if you go into crystallography, but do keep in mind that it's not easy, especially if you're working with membrane proteins. So trying to get these two different structures where molecules can bind and unbind is kind of tricky. And there's a bunch of different things you can try, like developing antibodies that bind to different states and then crystallizing the protein with the antibodies. So it's a very subtle change that you can detect, but it can have huge downstream effects. So when epinephrine binds on the outside, we see this movement in TM6 that will lead to the release of the G protein. The G protein is actually a heterotrimer consisting of an alpha, a beta, and a gamma subunit. When it gets activated, it actually gets released or dissociates from the membrane protein receptor, and the G alpha picks up a GTP molecule. The G alpha subunit is also lipid anchored, as you can see in this image, and in the GTP bound state, it can then travel in the membrane and interact with an enzyme called adenylyl cyclase. Activated adenylyl cyclase uses ATP as a substrate, and it creates a molecule called cyclic AMP, where you lose two of the phosphates on ATP and create a cyclic structure with the remaining phosphate. Cyclic AMP is considered a secondary messenger inside the cell that can activate protein kinase A. And protein kinase A is another enzyme that can phosphorylate many targets. Kinases generally phosphorylate other protein enzymes. So you can see how we can have many cyclic AMP molecules being made and then many pKa's activated and lots of other enzymes being phosphorylated, generating a bunch of downstream responses. This image is only showing one activation, but you can imagine that 10 effector enzymes can be activated and then 100 molecules are made, eventually leading to the breakdown of glycogen and fat to generate energy. Now, you may already know that epinephrine is considered the flight or fight response hormone, so this signaling pathway is what's going to give you the energy to run away or whatever else you need to do. I mentioned cancer, and RAS proteins are probably one of the most important oncogenes involved in cancer. The G proteins that we just talked about are large GTPases, they are heterotrimers, whereas RAS proteins are actually small GTPas. They only have a single subunit and combine GTP and hydrolyze it to GDP, so that's why they're called GTPas. A's just denotes that enzymatic function. When it gets turned on, it has GTP bound and acts like a molecular switch, allowing for signaling to occur. Now, 30% of all cancers are actually driven by point mutations in this protein. So these mutations will lead to the protein being stuck in the on state. The defect lies in GTP hydrolysis, so that's why they're called GTPases, meaning that they're constantly signaling. So they don't have the ability of hydrolyzing GTP to GDP and turning themselves off. Okay, and the downstream effect of this is cell proliferation, or grow, and the formation of tumors. We've actually known about this protein for 30 years, but it turns out we still don't have a drug that can target it. People have been trying to develop small molecules against it, but one of the problems is that it really doesn't have any deep pockets to bind a drug. If you were to look at the cell surface representation of this enzyme, the only deep pocket you would find is that GTP binding pocket, and it turns out that GTP has a really tight binding affinity. So it's really hard to get in there and disrupt it or compete for binding. So hormone signaling always occurs when you're talking about membrane proteins on the extracellular face, and this will induce a conformational change across the membrane, in this case for GPCRs, releasing that G alpha subunit in the GTP bound state. We then have adenylyl cyclase, which is the effector enzyme that will produce cyclic AMP, a secondary messenger. And then cyclic AMP can activate other enzymes like protein kinase A, which will transfer phosphates from ATP onto other enzymes to either activate or inactivate them. And of course, regulation of this cascade is very important. So we need to have lots of other things happening, either disrupting the binding interactions, metabolizing and breaking down these molecules, or dephosphorylation as well as protein degradation in order to turn off these pathways. Let's just switch gears. In the lipids lecture, I did mention that they can play a role in signaling, and there's an enzyme called phospholipase C that when activated cleaves phosphatidylinositol 4,5-bisphosphate. and it'll cleave at the bond between the phosphate and the glycerol backbone, producing diacylglycerol, so glycerol with those two acyl chains, as well as inositol 1,4,5 triphosphate, or IP3. These molecules can then act as secondary messengers, resulting in either the release of calcium ions, so that IP3 will bind to an IP3 receptor in the endoplasmic reticulum, open up that channel to allow calcium to leave the endoplasmic reticulum, and that calcium can actually activate protein kinase C, which is also activated by diacylglycerol in the membrane. This will facilitate its activation, and as the name suggests, protein kinase C can phosphorylate other enzymes to activate or inactivate them in the cell. Another way phospholipids can be involved in signaling is through the creation of eicosanoids. This is a large class of signaling molecules in the cell, and they're created from arachidonate. The release of calcium in the cytoplasm can also activate phospholipase A2, which cleaves the ester bond at the C2 position, releasing a fatty acid known as arachidonate. Arachidonate is a substrate for lipooxygenases that make leukotrienes, and these are a type of eicosanoid, but it's also the substrate for prostaglandin synthase, which also goes by the name cyclooxygenase or COX. Now, this is the third time we've seen this enzyme. First was in the fundamentals interaction lecture, and then in the membrane protein structure lecture, where we saw that it's anchored to the membrane by an amphipathic helix. It's also the enzyme that's targeted to alleviate pain and inflammation. When it produces prostaglandin H2, this can be used to make other eicosanoids, so the prostacyclins, thromboxanes, and other prostaglandins, and these all signal for pain and inflammation. The last type of membrane signaling we're going to look at are the receptors that have enzymatic function. You've probably heard about tyrosine kinases in your biology classes, and as the name suggests, binding of ligands leads to the phosphorylation of tyrosine residues. These receptors typically only have a single TM spanning the membrane, and upon ligand binding, two receptors come together and dimerize. They may also already be a dimer, and it's the conformational change upon ligand binding across the membrane that leads to the activation of an intracellular domain that's a kinase and can catalyze the phosphorylation of its partner. These phosphorylated tyrosine kinase domains can then catalyze the phosphorylation of various other target proteins. You're probably going to come across a lot of these types of signaling pathways in your other courses, so the epidermal growth factor, the JAK-STAC pathway, and the insulin signaling pathway is going to be very important in our metabolism lectures. So this is another type of signaling pathway that still involves a conformational change, transmitting the signal across the cell membrane. This slide is just showing you the changes in the insulin receptor. We've already talked about insulin, so it's a polypeptide-based hormone that has two chains bound by disulfide bonds, and it also binds to the receptor on the outside of the cell. Binding of one insulin molecule occurs at the interface between the alpha subunits, which are also known as the insulin binding domains. This activates the tyrosine kinase domain in the beta subunits on the inside of the cell, allowing for autophosphorylation. Triple phosphorylation, so phosphorylating 3 times, is actually needed to move an activation loop that can then allow other substrates, known as insulin receptor substrates or IRSs, to be phosphorylated in signal cascade. If you look at the hormones insulin and epinephrine, insulin signals for glucose uptake and use, while epinephrine can signal for glucose production from glycogen. So in essence, they are competing hormones, and you don't want them both working at the same time. This is where signaling starts to get very complex, and you can find maps on the web showing you the interrelationships between signal cascades. But when insulin is present, this can actually turn off epinephrine signaling. One of the insulin receptor substrates is another protein kinase B, and PKB can actually phosphorylate the beta-adrenergic receptor. When the beta-adrenergic receptor is phosphorylated, this actually signals for its internalization and degradation. So if the beta-adrenergic receptor isn't actually at the cell surface, GPR signaling by epinephrine can't occur, and insulin signaling will allow for glucose uptake, turning off glycogen breakdown. So obviously defects at any point in any of these pathways can lead to disease, and you are going to see this quite a bit in your upper year courses. We also have two upper year courses that deal with drug design and signaling. And there's lots of different conformational changes as well as types of post-translational modifications that'll be important for the activation or inactivation of different receptors and enzymes in these pathways. If you have mutations in any of these enzymes or receptors, that can prevent ligand binding as well as any protein-protein interactions that are needed for these signal cascades to occur. So it really comes down to knowing the structure of these membrane proteins in order to understand their function and be able to design different drugs that can bind and inhibit or even stimulate the proteins involved in signaling in order to modulate the different cellular responses. So the key take-home messages really are that signal cascades will amplify the original signal that occurs through binding to a membrane protein receptor. These receptors will transmit the signals across the membrane based on a series of conformational changes. And when we're talking about the non-steroidal receptors, they are integral membrane proteins where some of them only have a single transmembrane segment, whereas others are multipass. There's going to be lots of enzymatic reactions that happen in the signal cascades, and we create a variety of different secondary messengers that will act within the cytoplasm of the cell. Obviously, we do need to turn off these signal cascades, and that's going to be just as important for your physiological response. This is kind of an open-ended question, but if you were to target a specific GPCR and its function, where would you introduce a single amino acid mutation in order to modify its ability to activate its signal cascade? That's it for this week. Check out the Brain Break questions and make sure you review for the quiz this weekend.